Okay, hello and welcome to today's webinar, everybody, titled Using Single-Cell Multiomics to Analyze the Immune System. Before I pass over to our speakers, a few quick things from me. Questions for speakers. Feel free to ask questions whenever you like during the talks using the questions area on the right-hand side of your screen. Try to avoid using the chat area for speaker questions. So try and use the questions area if you possibly can. You can also upvote other people's questions. So feel free to go in and see what other people are asking. And if you want the speakers to answer that question at the end, then give a little upvote. We should have plenty of time for Q&A at the end when we'll bring all speakers back on screen to answer your questions. On demand as well. If you miss something one of our speakers said, or if you have to leave early, the webinar is available on demand. So you can watch the webinar back whenever you like over the next few days and weeks. Just use the link in your registration email, or go to our website at frontlinegenomics.com and you can see all of our webinars and you can click back in on this one. The on-demand version becomes available just about a minute after this live broadcast ends. So you can even hang about at the end and go back if you would like to. And if you want one of your colleagues to watch, they can also register for this webinar after it's finished today. So they can see the on-demand version whenever they like. So on to the webinar today. Today we have two fantastic talks from Anke and Dejay. But first, I'm going to hand over to Katerina from today's webinar sponsor, Singleron. Over to you, Katerina. Just going to bring
Thank you very much. Good morning, everybody. I'm very happy to have you here. I will just have a couple of slides explaining you the background of the technology for single-cell sequencing that we are developing. And then in a few minutes, I will hand over to Dr. Anke's Fenrich talk. So Singleron Biotechnologies is a company that specializes on single-cell sequencing, and we are currently present around the globe. So our European headquarters is located in Cologne, Germany, but we also have offices and labs in the US, Singapore, and China. So let me briefly tell you why we think single-cell sequencing is something that your research can benefit from. If you are already familiar with the bulk RNA sequencing, you probably know that in order to get gene expression information from your cells, you extract your RNA from the piece of tissue. And if this tissue contains different types of cells, in the end, you end up with the average gene expression profile from the entire sample. And this might not be optimal, especially in cases when your cell populations are very heterogeneous in your sample. And for this particular reason, the single-cell technologies have been developed. So the main difference between the bulk and single-cell sequencing is that in single-cell sequencing experiment, you start with your individual cells. You start with a single-cell suspension, and then you get the gene expression information for each of the cell and each of the cell type independently from each other. So basically, you end up with a lot of data that you can analyze then in any way you find to be appropriate. Now, how do we do that? So the advantage of Singleron's technology is that we have a chip, microfluidic device, that can be easily operated manually. So in order to start with your single-cell sequencing experiment, you don't have to buy any kind of expensive instrument dedicated for single-cell sequencing. You can simply operate this microfluidic chip with a pipette, as you can see in this picture here. So the chip, the scope chip, as we call it, is of a size of a microscopic slide. You can easily put it under the microscope and take a look how your experiment is progressing. Let me briefly explain how this scope chip works. So this device is covered with microvels. There is approximately 150,000 microvels in the standard configuration. And you use one of these openings on the chip to load your single-cell suspension onto the chip. So the cells settle gently by gravity into the individual microvels. And then you simply add your barcoding beads, and the cell and the bead would meet in the individual microvel. So once this happens, we can capture the mRNA from these individual cells onto the barcoding beads. So the beads themselves are covered with oligonucleotides, and they also have the poly-T tails, which capture the polyadenylated mRNA from the cell. So you can also see it on this picture on the lower portion of the slide. The bead and the cell come together, and then we simply capture the mRNA onto the barcoding beads. Afterwards, we take the beads with the bound mRNA from the chip, and we process with the reverse transcription, cDNA amplification, and the library construction. So in the end, you get sequencing-ready NGS library. So what we do in single run can be separated in two different groups. So we offer instruments, kits, and software for single-cell analysis, which you can do yourself in your own lab. You don't need to have a dedicated instrument for that, but if you want to, we also offer automated solutions to help you to automatize your workflow and save some time. So this is for those of you who would like to do these experiments themselves in your lab. For those of you who might be interested in outsourcing your single-cell workflow, we also offer service. We have a service lab located in Cologne in Europe and also in Singapore. And we accept the samples of the either fresh tissues in the preservation buffer or the frozen tissues, and we do the whole workflow ourselves in our lab. And we already have plenty of experience processing different types of tissues. So far, we have worked on more than 2,000 different research projects. And as an interesting fact, we have sequenced more than 320 million individual cells. And lastly, I would like to touch upon our multi-omic capabilities. So not only we offer kits for single-cell RNA analysis and the gene profiling, we also have other multi-omics products that enable you to do, for instance, the profiling of the VTJ sequences of the T and B cells. With our kits, you can also profile transcriptome and combine it with target sequences. So this is, again, on the single-cell level. You can, with our products, you can look at the transcriptome with temporal resolution, and you can also detect transcriptome with cell surface glycosylation, as you will hear later in one of our talks. So thank you very much for your attention, and without further delay, I will hand over to Dr. Fenrich to tell us more about her research.
Hi everyone, can you hear me? Because I can't hear anything. You can hear me. Great. I will start. Okay. Okay. Thank you very much for this invitation, and I'm happy to present our data regarding the single-cell RNA sequencing from nasal biopsies from symptomatic post-COVID patients. So just let me start with an introduction into the topic. As you all know, the SARS-CoV virus is the agent that causes the COVID-19 pandemic, and till now more than 600 million people confirmed the infection with COVID-19, and so far at least about 6 million people have died. As we all know that SARS infections are very heterogeneous. In some patients, SARS causes a multi-organ syndrome, but we also know that most patients recover completely. However, some patients turn to get more severe symptoms. Whereas we can see here, for example, after 4 weeks, a lot of patients will recover within the 4 weeks, but in a substantial number of patients, various long-lasting symptoms have been described. These long-lasting symptoms, for example, for over 12 weeks, we will now refer as to the post-COVID syndrome. Also for the post-COVID syndrome, we know that this syndrome has various symptoms without a cure yet, and some of which of these symptoms we also have, or these also occurred in patients of our cohort we were studying. And you can see here we have the data from our cohort we were interested in. You can see that the symptoms were varying between thought disorders, speech impairments. We know that patients will have different kinds of the fatigue, mental fatigue, as well as physical fatigue. We also know now that the post-COVID syndrome affects about 5% of all COVID-19 patients. And that's why it becomes very clear that the post-COVID syndromes has an enormous potential to become a global health burden. But so far, there is a lack of objective data to this topic, and there is a need for objective biomarker to stratify the risk for long-lasting symptoms. And therefore, we aim to use single-cell RNA sequences from post-COVID-19 affected or post-COVID syndrome affected tissue. Here, I want to present which kind of data we were working on, or what cohort we wanted to investigate. You can see, this is a big invitation from our colleagues in Kiel. We were working together, namely from Professor Barmer, Stefan Schreiber, Markus Weckmann, were colleagues that were working on this. We get access from to the patients from the NAPCON. NAPCON is in Germany this national pandemic cohort network. And here we get access to well-characterized patients. And here our colleagues have assessed the severity of the post-COVID syndrome, where they used detailed clinical data to develop the post-COVID score. The post-COVID score has 3 types. We have a mild form, a moderate form, and a severe form of the post-COVID syndrome. And we were working on patients either having moderate post-COVID syndrome symptoms or very severe post-COVID syndrome symptoms. Here you can see what we, which other comorbidities or other features we have in our data set. So you can see depicted in blue patients for moderate post-COVID syndromes, and in red patients with the severe symptoms. You can see that the patients were not varying that much in the BMI, not that much in the age. You can see that half of the cases were vaccinated and the other ones not. So there's not that much high heterogeneity in this data. So now I wanted to show you how we prepare the cell libraries. So we wanted to use or wanted to aim to perform single cell analysis also in a routine clinical setting. And as you all know or can imagine, that using this in a routine setting is often not that easily because it's not that common that single-cell sequencing is used in the context of patient stratification in such a clinical setting. In addition, before working with this single run, there was the need, or we have really stringent requirements for fresh sample preparation, which is also very hard to meet in a hospital setting. But these restrictions have been overcome by improvements on the sample and the library preparation chemistry and the workflow that the company Synchron offers. And therefore, you can see here a workflow how Synchron prepares the libraries and then send it away for sequencing. And by using their approach, we were able, in the end, to sequence up to 25 samples, and we were analyzing the single-cell data set with the state-of-the-art bioinformatic tools, and we could end up with about 35,000 cells, and the transcriptomic profile of these cells could identify 18 distinct cell clusters. And after looking for population-specific or cell-specific markers, we were able to identify the different cell types within this data set. And you can see that the cell types are, for example, here the epithelia cells, the ciliated cells on the left hand and on the right-hand side. You can see that we have found, for example, basal cells. We can see an immune cell population, and so on. As a next step, we were now asking, is there or can we find any difference between now this moderate and the severe patients regarding their gene expression profile? And indeed, we can see, I can spoiler this, what we see here is a pathway analysis where we compared the gene expression of moderate patients with the gene expression of severe patients. And here you can see on the right the pathways we are looking on, and you can nicely see that we see a shift in pathway activity, or pathway activity which leads to the gene expression, and we can see that in the severe patients we can see gene expression activation via TNF-alpha, NF-kappa beta, TGF-beta, and so on. And as a next step, we were now asking, because we have now the single cell resolution, we know the different cell types among our data set, we were now asking, are there specific cell types involved in this difference between moderate and severe patients? Here you can see we were looking or it's a very broad look. We will only comparing the frequency of the different cell types between moderate and severe patients, and you can, this is shown in this bar plot here. You can see, for example, for the basal cells that we have regarding the severity, we have an increase of cells. Regarding the ciliated cells, we can see that the ciliated cells get decreased from up to 20% in the more severe the post-COVID score gets. And we can also see an increase in basal cells. But now is the question why we do see this increase of basal cells and the decrease of ciliated cells. Therefore, we were interested, is there a difference between, here on the left we have our ciliated cells, and here on the right we had our basal cells. Is there a difference in the differential in the gene expression regarding to the post-COVID score or the severity of the post-COVID syndrome? And therefore we performed a differential abundance analysis. And from this analysis, you can nicely see that, for example, in the red group, so the red dots implies that, for example, we get, or we have a differential gene expression within the basal cells, also with the immune subpopulation. And for the more severe samples here on the left, it's depicted in blue that also the severe samples are showing a differential abundance, but here only in the ciliated cells. As a next step, we're now focusing, or we're now comparing the pathway activity, or the gene expression that was affected by the pathway activity, comparing moderate and severe patients, but now we were really comparing the different the different cell populations. So we have here a heat map. I think you are familiar with looking on heat maps. So this means the more red, the more activated a pathway was, or the more activated the pathway or the gene expression within the pathway. You can see here as a rows the different populations we identified so far, and you can nicely see now which pathways were affected compared from moderate to severe symptoms. What now comes a little bit into, or was some kind of eye-catching, is, for example, here the immune cell compartment. You can see that there are a lot of pathways are very upregulated. You can also see here the basal cells immune-influenced subpopulation that really shows an upregulation or an activation. But this is some kind of eyeboarding. We couldn't really distinguish between the moderate and the severe patients. Therefore, we're looking only on the immune cell population. So to say, we subset our data set on the immune cell population. And now, in regard of this immune cell population, we were comparing the moderate versus the severe patients. And here you can see this pathway activation again, or how the pathway activity was displayed, and you can here now see for the immune cell for the immune cell population that we have an activation of NF-kappa beta and TNF-alpha, and that we have a downregulation, for example, for TGF-beta and the P53 pathway. As one step now, we can, as we now know that, for example, the TNF-alpha and the NF-kappa beta pathway is activated, we also can take a closer look to the genes that were involved in this pathway, right? And you can see here for the left side, we were looking in genes in the TNF-alpha pathway, which genes were upregulated or not, and we can see it also from the NF-kappa pathway, which genes are involved and which genes are indeed up or down regulated. So to sum it up at this point, what we found so far is that we now could identify, or we isolated about 35,000 cells. We could identify 18 distinct cell populations, and there are a lot of known cell populations among them, like basal cells, ciliated cells, and immune cell populations. We could also show that that the severity of the post-COVID syndrome is in association with a decrease of ciliated cells and an increase of these undifferentiated basal cells, which in turn then was associated, as I showed you before, with an increased TNF-alpha, NF-kappa B response, or higher activation of these pathways. And maybe we can say that the TNF-alpha response signature maybe suggests this increase of this uncommitted basal epithelial cells, which in turn might interfere with a proper mucosal barrier function. What I showed some kind as an outlook here is we have done some pseudotime analysis where we compared the moderate patients with the severe patients, and from this pseudotime analysis we really have to run this again in more detail and with the higher stratification, but you can see that we here for the more severe patients we have more branching points that maybe gives you an advice for developmental arrest. And now I guess the most exciting question is now how we can verify our suggestions. As next steps, now we plan again to collect some patients' material. We will take nasal biopsies. We will do some cell cultures. We will confirm our findings also with some bulk RNA sequencing data as one branch, and as the second branch we will perform air-liquid interface cultures and will then add TNF-alpha and will then perform RNA sequencing and single-cell sequencing again. For the single-cell sequencing for this air-liquid interface cultures, we are working again together with SingleRun, and we are really looking forward for the results that were coming up. And with this, I wanted to thank you for your attention. I wanted to all these colleagues that were involved in this project. This is my working group where I'm working on. It's the Medical Systems Biology group in Lübeck. I wanted to thank the team from the Single Cell Core Facility and our collaborators also, from Kiel and Lübeck. And thank you very much for your attention.
Thank you very much, Anke. What an interesting presentation. Anke, if you wouldn't mind muting, if that's okay for the time being. Thank you so much. So our next speaker is Professor Jie Li of the Nanjing University in China. Jie, I'm just going to bring up your slides. Just give me one moment.
Oh, could you hear me?
Anke, I'm so sorry, Anke. Would you mind just muting your microphone? Just before Jie begins. Okay, let me bear with me, Jie, for a second. I'm just going to try and mute Anke. There we go. Over to you.
Okay, could you hear me?
Just about. Could you try speaking again? I think your microphone is coming in and out a little bit.
Okay, could you hear me now?
That's better. Yes, thank you.
Okay, thank you. Okay, it's my great honor to present here. invited by Singaron and Frontline Genomics webinar. So today I'm going to talk about like decoding the sweet information on the cell surface. That's the chemoenzymatic measurement of cell surface Glycan in single-cell multiomics. As we all know, cell surface glycans play an important role in cell biology. So almost all of the cells, mammalian cells, are coated by glycans, like O glycans and N glycans. Glycans play important roles in like cell interaction, like nerve conduction. So in today's topic, I want to talk about its role in immune response. So to this point, glycan-3, as we know, is secreted by cancer cells, can binding N-glycans of TCR on T cells to inhibit T cell activation. So the human system usually take glycans on cell surface to do a lot of things in immune system. So as glycan information is very important and very heterogeneous in the immune cells, so we want to decode this information in single cell level. Actually, in the past few years, like researchers from Australia and Japan, they do a lot of single cell glycan detection. So they use acting in this single cell detection method.
Jay, I'm so sorry to interrupt you. Your microphone is cutting in and out, so we can't. Is there anything you can do at your end just to help with that?
It's now okay.
That's loads better. Thank you so much. Thank you.
Okay. So in their works, they use lectin for glycan staining on single cells and convert this information to the like sequenceable DNA information or the metal staining for CyTOF. But it is to there is a very important information here. The lectin staining is multivalent and may perturb the transcription of the cells. So actually, there are two methods for like glycan detection on cell surface. One is lectin staining and one is chemoenzymatic labeling. This method is developed by chemists and chemical biologists. As we all know, Caroline Bertozzi won the Nobel Prize last year, and she is a pioneer of chemoenzymatic labeling on cell surface. So this chemoenzymatic labeling is stable covalent labeling, and it can recognize glycan unit, not just the uniform structures, and it is monovalent labeling, not like the lectin is a multivalent binding, usually cross receptor on cell surface to induce signaling transduction. So it seems this method is very compatible with like the RNA-seq or single-cell RNA-seq because it has very small influence on the signaling pathway. So in this work, we focus on LACNAC unit because LACNAC unit play an important role in immunity. As we know, the cells are coated by the glycans in very complex structures, but all the building blocks contain this disaccharide called LACNAC unit. It's like this chemical structure. So in previous report, Glycan-1 bind to T cell surface polylacnac to inhibit the T cell function. In another work, people found that the N-glycan branching controls the differentiation pathway of CD4 T cells. When the glycan branching, the Treg is more, and when the glycan branching is low, Th17 cells is more. So it is very important to detect the LACNAC unit on cell surface. a very genius work from the NYGC. They developed the SciSeq, shows an example of single-cell multi-omics. They introduced the epitope information into the single-cell RNA-seq that we can detect the transcriptome with glycan with epitope information together on usually different on different platforms like the Synchron and 10x. All of these the commercial platforms works with these sites like epitope and transcriptome detection in single-cell multi-omics. So our workflow just like we are inspired by this the SciSeq technology. We just use.
I'm so sorry to interrupt again. That problem is back again. Is there anything you can do to?
Is that okay now?
Yes, it is. It seems to be. Yes, thank you.
Okay, sorry, because we have some block of the internet. Sorry. Okay. Yes, in this platform, we just inspired by their work. They use antibody DNA conjugate to convert the epitope information into sequenceable DNA information. In our platform, as I introduced above, we can use chemoenzymatic labeling to put a glycan link ssDNA to the LACNAC unit on cell surface, and then use these commercial like single-cell sequencing platform. We can detect the glycan information as well as the transcript information together. And then we can detect their correlation or their relationship between the glycan information and the genes. In our previous work, we developed a Focal T enzyme-based chemoenzymatic method for LACNAC detection. We can use 1,3 Focal T transferase from the H. pylori. It can put the fructose linked ssDNA on the LACNAC unit on cell surface. So it helps us to put a ssDNA tag, like use Synchron rules, put the UMI and put the sequenceable the barcode on the DNA on the sugar. So then this enzyme transfer this DNA barcode.
Could you hear me now? So it looks like it's disconnected again. Oh, okay. So before we like put this method to the single-cell sequencing, we use the glycan chips to confirm this 1,3 Focal T can label most LACNAC unit. Use these glycan chips. So in this data, we can find that we can label most of glycan structures like bearing the LACN unit. Only like the LACNAC unit with some steering hindrance. So it tells us we can label most of the glycan unit, LACNAC unit on cell surface. So this fluorescent imaging data was further confirmed by the mass spectrometry. So we can confirm the ssDNA tag are linked to the cell surface LACNAC. So when every molecular information is confirmed by our chemical or detection method, we move to the next step. We put this tag on cell surface and then use the commercial single-cell platforms to detect cell surface LACNAC simultaneously with endogenous transcript. So every platform works, single 10x works identically. So we can detect these transcript and use a transcript to discriminate these cell lines. And on every cell line, we can show the quantification of the LAC-NAC is identical to the flow cytometry result. So it says that we can detect LAC-NAC on cell surface quantitatively. Next step, we want to confirm the LAC-NAC detection has minimum influence on the transcriptions, because as I mentioned above, the lectin staining or other glycan staining method may cross-link cell surface receptors to induce the signaling pathways and change the transcription inside cells. So here we use a spleen, and we split these spleen samples in splenocyte into two parts. One is untreated, one is treated with our chemoenzymatic LAC-NAC labeling reagent. So we can find that these two samples are most identical, and the LAC-NAC labeling has minimum influence on the transcriptome on each cell type, like T cell, B cell, and NK cell, all is fine. So this data is from the syndrome the collaborators. Next, we want to combine LAC-NAC-seq with Cy-seq, because we want to detect cell surface glycan information with cell surface epitope information and the endogenous transcription information. So actually, it is very easier to combine these two platforms. We buy the ssDNA tag antibody from BioLegend, and we make the GDP focus ssDNA, put the enzyme and the GDP focus ssDNA and with this antibody together to incubate with these cells. So in this experiment, the human PBMC was treated with CD3, CD28 antibody to stimulate, and there is another sample with untreated to detect the different T cells or different immune cell phenotypes and see what glycan formation on these different cells. So as we can see here, we can show the PBMC has different populations here, and the CD4, CD11C, and the CD19 can easily use the epitope information to identify each subpopulation. And then we can merge the LAC-NAC information into this UMAP picture, and we can identify different LAC-NAC expression levels in single cells in different immune subtypes. So we also use the trajectory along the timeline to see the T cell differentiation, and we find the T cell activated with effector phenotype has the most increased LAC-NAC level. Actually, there is previous work use the PHA staining to detect the LAC-NAC linked to the manners on cell surface, and they found that the PHA staining is higher in the clone expansion T cells, but in the less expanded T cells, the PHA staining level is low. That is to see the PHA staining or the LAC-NAC linked to the manners is associated with the T cell clone expansion. Because we are detect similar things, like they detect LAC-NAC linked to manners, we detect LAC-NAC. So we also compare these with their work. So we combine the LAC-NAC-seq with single cell TCR-seq That is to see we can detect the LAC-NAC information on the cell surface, the transcriptions, and the TCR clone informations. So we perform this with the syndrome the 10x platform, but later we use this platform, this system with the syndrome platform. They can also detect the TCR information simultaneously with the LAC-NAC. So it is very interesting here is that in our system, we detect the tears from the breast cancer, tumor breast cancer samples. So in our experiment, we could only find the effector CD8 T cells has significantly upregulated LAC-NAC level. As the PBMC sample, we activated with CD3 antibody. But when we correlate this information to the TCR clone expansion, we can see some trend here, but it is not very significant. So that is to see chemoenzymatic LAC-NAC labeling is totally different from the PHA staining because they recognize different patterns. So later we use in vivo assays to confirm this result. We use a Listeria infection model that to stimulate OT1 T cells in naive mice. So when we transfer 1,000 OT1 T cells into this recipient mice and use 2 Listeria strands, one with N4 strong TCR engager, one with T4 is a weak TCR engager. As shown here, the N4 stimulate a lot of OT1 CD8 T cells here. It represents that the T cell is very clone expanded here. Not clone expanded, is very expanded here. But T4 only induce a very small number of proliferation or expansion. However, when we gated these cells on the activated subpopulation, they show identical LAC-NAC level. That is to see this LAC-NAC is correlated with T cell like functions, not the clone expansion levels. Because the glycan is synthesized inside the cells and transport with glycan proteins to the cell surface. So the metabolic pathways contribute to this N-glycan synthesis inside the cells. So we want to figure out what contribute to the LAC-NAC level on T cell surface, especially in the effector T cells, YZ up the highest level of LAC-NAC. So we use the GSEA analysis, the GSEA analysis to show which metabolic pathway is most correlated with this LAC-NAC level. The GSEA data shows that LAC-NAC in CD8 T cells are most correlated with glycolytic activity, not oxidative phosphorylation or TCA cycle. Actually, we then analyze a public database to find that the LAC-NAC level is very correlated with the glycolysis pathways. This uses pathway activity correlation analysis. So as we know, in the N-glycan synthesis pathway, it is coupled with the glycolytic pathway. So the HK1 and GPI converts the glucose to the F6P as the important starting material for the glycolytic pathway and the LAC-NAC synthesis pathways. So as we analyze the public database, we can find the effector CD8 T cells most upregulate HK1, GPI1, and LDHA that is correlated with glycolytic activity. In the same time, they also most upregulate like B4GLT1, B3GNT1, and MGAT1. This is our glycosyltransferase responsible for the LAC-NAC assemble on T cell surface. So our data suggests that CD8 T cell express the highest level of LAC-NAC due to its very high glycolytic activity. So that is to see the LAC-NAC level in on CD8 T cell surface might be a marker or an indicator of its metabolic activity, especially the glycolytic activity. So we next use the inhibitor to confirm this conclusion. We use 2DG to shut down the very important glycolysis pathway to inhibit the converting from glucose to F6P. So as we can see here, when we add the inhibitor, the T cell activation is totally blocked, and the LAC-NAC level is very low than the untreated group. So this data suggests that glycolysis affects T cell activation and LAC-NAC upregulation. So due to this finding, we think that maybe glucose, the actual glucose or high glucose supplement may promote the CD8 T cell proliferation during activation. So when we activate T cells, we add extra glucose, and we found that this actual glucose supplement significantly promoted CD8 T cell proliferation during activation. So this high glucose supplement may boost the proliferation of CD8 T cells, especially the CAR T cells, when we in vitro expansion. So this may be a very good finding for the translation that discovered by single cell multi-omics with glycan and transcriptome information. So at last, I want to introduce our chemical single cell multi-omics study. So in previous study, we have seen the power of combining the epitope or glycan detection with the transcriptome, because they can link in the chemical information with the transcription information in single cells. That is to see we can know how the chemical signals or chemical modifications are regulated by the transcription or by the enzyme inside a single cell. So actually the cell is built by protein, by lipid, glycan, and by metal ion. So we can image that if we can correlate these chemical signals or chemical element or chemical modification on biomoleculars together with the transcription, reflect the physiological function of the cells, maybe we can get very exciting new insight in single cell level. Let's summarize my talk here. First, we found that we can combine chemoenzymatic labeling with single cell sequencing. And second, we found that the LAC-NAC-seq integrates the analysis of LAC-NAC and the transcriptome of T cells at a single cell level. Based on this technique, we found that cell surface LAC-NAC is significantly upregulated in activated CD8 T cells, but not associated with TCR clone expansion. It only related to the T cell function. So at last, LAC-NAC level is positively correlated with glycolytic activity of CD8 T cells at all stages that is discovered by this single cell detection technique, and may have some insight for the CAR T therapy, the in vitro expansion protocol. So let me thank my collaborators from Scripps and Nanjing University and my student. Okay, that's all. Any questions?
Thank you very much, Jay. What a great presentation with lots of really interesting data and some questions as well. I can see that Anka and Katerina have been busy answering questions in written format. So if you did have a question earlier on, please do go back to the questions tab and take a look for the answers. Okay. You can see the answers on the on-demand version, but Jay, we do have some questions for you, if that's okay. And I'm going to take them in the order in which they were asked, and I'll leave your slides up in case you need to go back. So this question is from Apona Sikar. Are LACNAC units present in both O-linked and N-linked glycans?
Yes, it can detect LACNACs on O-link and N-link. Both is fine.
Thank you. And then I have two questions from Alessia Buratin. The first one is, the multi-omics data were obtained in a pairwise manner for each cell? Question mark.
Pardon me?
The question is, I guess I'll ask it in a slightly different way. Were the multi-omics data that were obtained, were they obtained in a pairwise manner for each cell?
Yes.
Yep. Okay, wonderful. And a final question then from Alessia as well. Which statistical model did you use to integrate the multi-omic layers?
Could I find these questions in the question table?
Yeah, absolutely. So if you go to the questions tab on the bottom right, and it should automatically order by date, but you've got the option of order by. And if you order by date, it's the top question. It's a two-part question.
Oh, okay. I can see it.
Perfect. Thank you.
Okay, the statistical model is actually we do it by ourselves. We write a code for this. Yeah, we do the log on the LACNAC the data, and then combine with the transcriptome, because the LACNAC unit is much higher than the transcription.
Brilliant. All right. Look, thank you so much to everybody. I think that's all of the questions answered. Just to go back to our speakers, do any of you have any final closing comments or anything that you wanted to add that was quite difficult to type? No? We're all good?
I might actually add to how the chip works, because we had several questions on that. Basically, how is it possible that you get single cells in the chip, and how come that they are actually separated from each other? So basically, how the chip works is that you have plenty of microvels there. You have 150,000 microvels on the standard chip, and you only capture 10,000 cells. So majority of the microvels remain empty, and in the ones where there are cells, it's usually single cell per microvel. So this is how it works, and this is how we ensure the partitioning of the individual cells into the individual compartments. So I think this answers a couple of questions that people have raised.
Great. Thank you so much, Katerina. Okay, well, look, I think we'll close off the webinar there. Thank you for joining us today, all of you. Thank you for your questions, a lot of questions. So you've kept our speakers very busy, which is fantastic. Thank you to our speakers as well. I know we've braved some technical difficulties during this webinar, I think for all four of us, actually. So thank you so much for hanging on in there. I think you did a great job, and what wonderful presentations as well. For those of you listening, don't forget this webinar is available on demand, so you can come back and watch it at any time. And please encourage your colleagues to register. If you think they'll be interested, they can still register even though the live broadcast is about to end. That's it. Thank you again to our speakers. Thank you all. Have a lovely rest of your day wherever you are, and goodbye.
Thank you. Bye.